My name's Cindy Atwell, and I'm the Chief Business Officer at Precision BioSciences. So today I want to introduce you to Precision 2.0 and our new identity, as well as talk about ARCUS, our proprietary genome editing technology, and the ways that ARCUS is unique and differentiated. Additionally, we'll go through our programs, our wholly owned organic programs that we're advancing rapidly towards the clinic. And then we'll touch on very briefly some exciting new gene insertion data using the ARCUS gene editing technology. So Precision is a public company, and I will be making forward-looking statements today. So we completed our pivot from a dual platform company to a single platform company focused on in vivo gene editing. This was enabled by our strategic transaction with Imugene. Imugene will take forward Azer-cel, our lead allogeneic cell therapy program, as well as have taken on the cell therapy capabilities, leaving us with a very clear mission and a very clear focus on in vivo gene editing. Now, ARCUS is a sophisticated gene editing tool, and what I mean by that is there are a couple unique attributes that ARCUS has which really enable differentiation and differentiated products. That includes the cut, the type of cut, the size of the ARCUS technology, as well as the simplicity of the construct itself. So with regards to the cut, ARCUS leaves a 3' 4 base pair overhang. This is important because it drives homology-directed repair, which increases the efficiency of gene insertion as well as excision and perfect religation. Additionally, the size matters. ARCUS is about a little over 1 kb, so it can easily fit into LNP delivery or AAV delivery. So we can then deliver to the liver as well as tissues outside the liver. And lastly, ARCUS is simple. It doesn't require a guide. The recognition sequence, as well as the sort of gene editing machinery, is all encapsulated within one single protein, and a small protein at that. So what this really leads to is more defined outcomes and ability to target tissues outside of the liver as well. And when we think about applying ARCUS and our focus with ARCUS, whether it's through internal organic programs that we're bringing forward or through partnerships, we're really thinking about ARCUS with a more sophisticated edit. And what we mean by that sophisticated edit is depicted on this tree here. So we won't focus on liver-directed knockouts. Instead, we'll look at excision or elimination and insertion or even repair. So these are more difficult edits or complex edits, but ARCUS is really enabled to enable these type of edits because of that 3' 4 base pair overhang, that type of cut it makes. And you can see our pipeline here where we're really focused initially on our hepatitis B program as well as our mitochondrial disease program. Both of these are elimination programs, so we're essentially eliminating the virus in the case of the hepatitis B program or the mutant mitochondrial DNA in the case of our mitochondrial program. I won't have time to go through our partnered programs today, but many of our partners have applied ARCUS for gene insertion or excision, as you can see here. So now let's focus on our lead program, our hepatitis B program. Unfortunately, hepatitis is a leading cause of mortality and morbidity in the U.S. and globally, with globally over 300 million patients affected by chronic hepatitis B. It's an estimated 15 to 40% of patients will develop liver complications, liver failure, or even liver cancer, which really accounts for the majority of HBV-related deaths. Now, HBV is a wily virus in the sense that when it sets up shop in an infected cell, it hangs out, the viral DNA hangs out, the cccDNA, or covalently closed circular DNA, which is essentially the viral genome, hangs out in the nucleus of the cell, but it also inserts its DNA into the patient's genome. And so we have an approach which is differentiated, where we are basically eliminating the cccDNA, and additionally, we're knocking out the integrated DNA. And you can see here in an episomal mouse model that we inactivated the viral DNA and durably reduced S antigen by 96%. So it's important to note that this was sustained over the course of the study, that this reduction in S antigen as well as the viral inactivation or elimination. Also looking at a monkey study, again an episomal study, we saw that this Arcus nuclease eliminated or inactivated 80% or greater than 80% of the total viral DNA. Now, I should note that both of these studies were conducted with earlier generations of our Arcus nuclease, which is directed towards the HBV genome. We have since basically improved the HBV Arcus nuclease as well as the platform overall, leading to better efficacy and safety overall with our clinical candidate. Now, that data will be shared at a scientific conference later this year. Additionally, I would say we are on track for a CTA or IND filing by end of 2024. And we are aiming for a functional cure of HBV with this program. Turning to our second program, our primary mitochondrial myopathy program, or PMM for short, we just unveiled this last month. And this disease, mitochondrial disease, is actually one of the most common hereditary metabolic disorders. We are targeting a specific mutation, the 3243 mutation, and looking to alleviate or potentially even cure the myopathy or the muscle-related symptoms. Now, we all know from 8th grade biology that the mitochondria is the powerhouse of the cell. And the mitochondria actually have their own DNA, different DNA compared to the nuclear genome. There are about 100 copies of mitochondrial DNA per cell, and in the case of a 3243 patient, they have a mix of the mutant DNA, which isn't functional, and wild-type DNA, mitochondrial DNA, which is functional. Now, what that refers to is heteroplasmy, and what we're really aiming to do is essentially eliminate the mutant DNA and allow the wild-type mitochondrial DNA to repopulate the cell. And so that's actually what we see here in this in vitro assay, in this in vitro data. So on the left-hand side of the screen, we see the control with the orange, greater than 90% mutant DNA. So these are in vitro studies using 3243 patient cells. And when we apply the PBGene PMM Arcus nuclease, we see a shift in heteroplasmy, really shifting the DNA to the wild-type mitochondrial DNA. And what that results in is an improvement in energy production within the cell. Additionally, we have tested a surrogate in a mouse model as well through a collaboration with the University of Miami and Dr. Morris. And we see here, through delivery of AAV9, we're able to shift heteroplasmy in the mouse model across a number of different tissues. So this is an opportunity for a one-time administration and potential cure for myopathy associated with mitochondrial disease in these 3243 patients. Unfortunately, today, there are not many good options. Typically, the standard of care for this indication is a vitamin cocktail. So these patients are really looking for something to alleviate their symptoms. We are on track with this potential first-in-class opportunity for a CTA or IND filing in the 2025 timeframe. Now, just because of time, I will very briefly touch on some gene insertion highlights and some exciting data that was recently generated using ARCUS. So this is a true head-to-head comparison against CRISPR in vitro, and we were looking at ARCUS versus CRISPR, and we see a difference of about 17x in efficiency with regards to gene insertion. And the reason we think that there's this big difference, and through further testing have validated, is that it's the type of cut that the ARCUS nuclease makes. That 3' 4 base pair overhang really drives homology-directed repair, which lends itself well for gene insertion. Now, we see this carried forward through in non-human primate studies. So this is a study, a non-human primate study, where we tested and delivered the ARCUS nuclease to the liver via LNP and mRNA. We delivered the repair construct at about the same time via AAV. And what we've seen is about 40 to 44% gene insertion. This is, from what we could find in the literature, about 3x what we understand is the reported highest rate for the CRISPR-based approaches. So that 3' 4 base pair overhang really driving the homology-directed repair, even in adult non-human primates, so non-dividing cells. So the path forward is clear for precision. We'll drive our organic internal program growth through HPV and the mitochondrial programs with CTAs and INDs in the 24 and 25 timeframes, respectively. Future programs will look at those sophisticated edits and really applying ARCUS towards a sophisticated edit, creating a differentiated product. So looking at insertion or excision. Additionally, we're continuing to seek partnerships, seeking partnerships to apply this technology, again, for sophisticated edits, similarly to our Novartis and Prevail Lilly collaborations, as well as our EACare collaboration, looking at insertions, excisions, or even potentially repair. Additionally, I'd note that we're continuing to seek partnerships for our allogeneic cell therapy program, the stealth cell, PBCAR19B, as well as azer-cel for autoimmune indications. All right, thank you for your time.